Every time bacteria reproduce, the population doubles. So the end bacteria number equals the original bacteria number times 2 to the power of the number of divisions. Let's have a look at this over 60 minutes. So at 0 minutes, we have 1 bacterium. After 20 minutes, this has reproduced and doubled to give us 2 bacteria. After another 20 minutes, it doubles again to give us 4 bacteria after 40 minutes. Give it another 20 minutes and the population has doubled once again to give 8 bacteria after 60 minutes. So you can see that every time the bacteria reproduce, the population doubles, hence the 2 in this equation. You will be expected to use this formula to calculate the number of bacteria in a population after a certain amount of time if you're given the mean division time. So how do we use this formula in practice? The mean division time for bacterial population A is 20 minutes. If the observation begins with 1 bacterium, calculate how many bacteria will be present after 5 hours. So step 1 is to calculate how many times the bacteria divide in an hour. To do this, we just need to divide 60 minutes by 20 minutes, which is the mean division time, which gives us 3 divisions an hour. Next, we calculate the total number of divisions. So we've calculated that there are 3 divisions an hour. We can then multiply this by 5 hours. This is given to us in the question. And this gives us 15 divisions. So now we've got the total number of divisions. The next step is to insert the number of bacteria and divisions into the formula to calculate the number of bacteria at the end of the growth period. So here's the equation that we're working with. So end bacteria number equals original bacteria number times 2 to the power of the number of divisions. So in this example, it will be 1 times 2 to the power of 15, which equals 32,768 bacteria. You are expected to memorize this formula and be able to perform these calculations in the exam. Many bacteria reproduce very quickly, so populations of bacteria can rapidly become very large. The size of bacterial populations is often shown in standard form. This simple diagram shows a petri dish, and inside the petri dish is a bacterial growth. We know from looking at this petri dish that the bacteria have replicated, but how can we work out the size of a bacterial population? The mean division time for bacteria population B is 20 minutes. If the observation begins with 1 bacterium, calculate how many bacteria will be present after 10 hours to 3 significant figures. Present your answer in standard form. So, step 1, calculate how many times the bacteria divide in an hour. So 1 hour is 60 minutes, and we know that the mean division time is 20 minutes. So a simple 60 minutes divided by 20 minutes gives us 3 divisions an hour. Next, we need to calculate the total number of divisions. So we know from our previous calculation that there were 3 divisions an hour. The question is asking us to calculate how many will be present after 10 hours, so we'll pop that into this multiplication. 3 divisions an hour multiplied by 10 hours equals 30 divisions. So we now know the total number of divisions. How do we use this to work out the total number of bacteria present at the end of the growth period? Step 3. Insert the number of bacteria and divisions into the formula to calculate the number of bacteria at the end of the growth period. Here's the formula, so remember that the end bacteria number equals the original bacteria number times 2 to the power of the number of divisions. So in this case, it will be 1 times 2 to the power of 30, which gives us this value. You should be able to calculate the number of bacteria in a population after a certain time if you're given the mean division time like you are in this question. So now we have our answer, but you can see that this number is very large. How do we present this to 3 significant figures in standard form? For step 4, we need to convert the bacterial population size into standard form. So to do this, we need to identify the 3 significant figures within this overall number and convert it into the standard form. In this example, the three significant figures are the 1, 0 and 7 over here, and we can convert this into 1.07. To get to this figure from 1.07, we need to multiply it by 10 to the power of 9. This gives us the answer in standard form. Now, this conversion can be done on a calculator, and normally your calculator will automatically put whole numbers with many digits like this one into standard form. Only higher tier students should be able to express the answer in standard form for bacterial population sizes.
Thanks for watching. If you want to take your GCSE revision to the next level, head over to launchpadlearning.com and check out our smart learning platform that's been designed to get you top results in your exams. We cover your whole specification and make revision fun with interactive quizzes, easy-to-follow videos, and more. You'll be kept motivated by your own AI tutor who's here to support you every step of the way. To check it out for yourself, click here. Or click here to keep watching a selection of the videos from our full GCSE Biology course. See you there!